So I'm Dr. Amalia Gastaldelli, and I'm from Italy, the Pisa CNR, and I'm here today to chair this very interesting session, and we will talk about several things related to the MASH. So the first presenter is Professor Sonia Najjar, that is chair of the Academic Research Center Biomedical Sciences Diabetes Institute at the University of Ohio. Unfortunately, she had an injury, so she cannot be here in presence, but she sent her video. So we will start with that, and then we have the other two speakers.
Good afternoon, and sorry I'm not with you in person, but a fractured heel bone precluded my physical presence. But I'm very much wishful that I'll be able to convey the right message through this pre-recorded lecture. And the topic of the day is insulin resistance an important component of the pathogenesis of hepatic fibrosis. As you know, especially when we deal with hepatic fibrosis vis-a-vis MASH and how it progresses from MESL-D to cirrhosis and others. For example, you can start with simple steatosis, simple fat accumulation in the liver, that's followed by infiltration of inflammatory cells and collagen deposition to lead to a variable degrees of or stages of fibrosis. If uncontrolled, this would lead to cirrhosis and eventually HCC. So the question that we are discussing today, and the question that has not been totally resolved, is that, well, whereas insulin resistance is very important in the early part of the disease, like when there is metabolic dysfunction, is insulin resistance involved or implicated in the later stages when there is liver injury, hepatocytic injury? And this is very important because insulin resistance has been characterized as very important to understand the classical features of the spectrum of metabolic syndrome, such as obesity, dyslipidemia, and hypertension. And it's important that we try to figure out the role of insulin resistance in the newly added components, such as MESL-D, MASH, and atherosclerosis. And again, insulin resistance means different things to different people. To me, I have always given it a liver-centric view vis-a-vis, in light of the fact that we have been focusing on CCAM1. CCAM1 is a very important protein. It's very highly expressed everywhere, but most predominantly in hepatocytes. And given the role of hepatocytes in insulin action, insulin clearance, it was inevitable that we will investigate how CCAM1 can play a role, if any, in hepatic insulin action, and whether a decrease in CCAM1 or inactive CCAM1 can lead to insulin resistance and perhaps the different components of metabolic syndrome. And that's what has been the focus of my lab. So why is it so important? It's because hepatic fibrosis is on the rise. Hepatic fibrosis is on the rise in parallel to MESL-D, and which is very high in Southeast Asia and the Gulf countries and in Latin America, but North America is not being spared, and we are following the trend. So, indeed, hepatic fibrosis is on the rise. Maybe perhaps is that we haven't found a fibrosis-specific or fibrosis-targeted therapy, because maybe we don't understand its pathogenesis as well as we should. And so a lot of research has been happening in order to dissect out the molecular basis of the disease. But certainly, hepatic fibrosis increases the risk of, or is very high, increases the risk of mortality from liver-related dysfunctions. Some of the drugs that recently have been used is to we repurpose a lot of these insulin secretagogues and insulin sensitized drug to be able to control hepatic fibrosis, incretins, or especially here, as shown here in this more recent CellMet review, is that, as you notice, there are a lot of genes that are implicated in insulin action, lipid homeostasis, and more recently in thyroid. So in many ways, this has been, many of these drugs have reached clinical stages 3Bs, but 3B, but in a sense, we still haven't yet found the best therapy, and we haven't understood all the pharmacokinetics of these newly developed drugs. We're still where we were back in 2008, and the reason is that when you focus on insulin resistance alone, or insulin action, or incretins, it's very good. It may help a lot, and the mainstay of therapeutic modality remains lifestyle interventions. But you ignore and you exclude many patients who manifest inflammation and fibrosis without necessarily insulin resistance. To those, sometimes angiotensin receptor inhibitors or blockers have been used, but still a lot of unresolved questions remain in this respect. So obviously we joined the fray and we tried to use CEACAM1 as potentially, can we use CEACAM1 as a potential therapeutic target? To do so, let's understand if CEACAM1 plays a role in the pathogenesis of hepatic fibrosis. CEACAM1 is a substrate of the insulin receptor. It is mostly expressed, especially the long isoform, is expressed in the sinusoidal domain of the hepatocyte. That's its highest expression. And the long isoform contains this cytoplasmic site that has this tyrosine that gets phosphorylated by insulin as well as other growth factor receptors, such as EGF receptor and VEGF receptor. As you will see, and as I'll share with you in terms of mouse models, there has been some clinical studies in parallel to our findings, namely this study back in 2011 by a Southeast Asian pathologist that showed that hepatic CEACAM1 expression is decreased in the liver of patients that are obese with fatty liver, regardless of, independent of insulin resistance or diabetes. In subjects with insulin resistance and fatty liver, but not necessarily diabetic. That's in hepatics, in terms of hepatic CEACAM1 expression. More recently, in collaboration with Professor Macedo in Portugal and her lab, using 1,019, focusing on 1,019 patients that are involved in the PRIVA type 2 Portuguese cohort, we showed that not only hepatic insulin expression, but also circulating level of insulin decreases as the disease progresses from normoglycemia to prediabetes to type 2 diabetes, in parallel to reduction in insulin clearance, increase in fatty liver deposition. In collaboration with Drs. Aliza Rimpar and Sergio Duarte at the University of Florida in Gainesville, we also showed that hepatic CEACAM1 expression is remarkably lower in patients with MASH than in patients that don't have MASH, and that the CEACAM1 level decreases progressively as fibrosis stages advances from F0 to F3 to 4. We also showed that by focusing on databases that are available, single cell RNA sequencing, showed that CEACAM1 level decreases in hepatocyte, in mesenchymal cells like stellate cell, and in endothelial cells in patients with cirrhosis or advanced fibrosis as compared to subjects with non-injured liver. So we wanted then to use different mouse models to dissect out the specific role of CEACAM1 in hepatocytes, where it is mostly expressed, followed by LSEC, liver, the hepatic, or LSEC, the liver sinusoidal endothelial cells, followed by hepatic stellate cells. And we have published all of this. So we mutated or deleted specifically CCAM1 in these different cells in the liver and investigated what is the role, what is the effect of this deletion on A, insulin action, B, liver histology. And by so doing, we aimed again studying whether this implicates insulin resistance or not. Why do we care about insulin resistance? Well, because many of us know that insulin signaling mediates insulin action, but we also have to remind ourselves that this is regulated by how much insulin circulates in the blood. And how much insulin circulates in the blood depends not only on beta cell secretion of insulin, but also on insulin clearance, again, mostly in hepatocytes, and to lower extent in the proximal tubule cells of the kidney. And this balanced level of insulin that determines the insulin response in peripheral tissues like myocytes and adipocytes. So what have we shown? So at the cellular level, we have shown early on that CCAM1 being a substrate of the insulin receptor, mostly expressed in hepatocyte, that it gets phosphorylated. And by so doing, how does it get phosphorylated? In response to the acutely released insulin spikes coming out of the beta cell into the portal vein via the fenestrae of the capillaries surrounding the hepatocytes. And this will activate the insulin receptor, phosphorylate CCAM1. CCAM1 takes part of the endocytosis complex in order to stabilize it and increase the rate of endocytosis and targeting insulin to its degradation in the late endosomes. But for insulin to be degraded, insulin has to dissociate from its receptor, and the receptor recycles back to the plasma membrane. So to destabilize this complex, fatty acid synthase that's very highly expressed in the perinuclear region binds to CCAM1 on the phosphorylated tyrosine, takes it away from the complex, and that leads to a decrease in fatty acid synthase activity. What we have shown by so doing is that this is the mechanism via which the hepatocytes or the liver are protected against the high level of insulin in the portal vein. For example, since we know that the insulin concentration in the portal vein is usually at least minimum 2 to 3 times higher than the systemic circulation, we always question if insulin is always lipogenic, then the liver should be normally fat, and fatty liver is not a disease, given the fact that insulin is lipogenic. But consistent with the fact that the chronically elevated level of insulin lead to increase in the expression of lipogenic genes, indeed, fatty acid synthase expression is very high in the hepatocyte. But we all know that the activity under normal physiologic condition is almost zero. So what protects the liver against this otherwise lipogenic effect of the higher level of insulin in the portal vein are 2 things. The pulsatility of insulin, so acute rise of insulin that reach the insulin receptor, which then will activate CCAM1, activate the receptor and phosphorylate CCAM1. And the other protective measure is that CCAM1 increases the rate of endocytosis, mediates this clearance to decrease the level of insulin, and at the same time mediates the decrease of fatty acid synthase activity by acute insulin. So insulin acutely suppresses de novo lipogenesis, just like it suppresses gluconeogenesis, and as such, this promotes the idea that insulin resistance, at least hepatic insulin resistance, is not selective. That it includes the inability of insulin to suppress both gluconeogenesis as well as de novo lipogenesis, in agreement with the most recent work that shows that hepatic insulin resistance is not selective, especially in patients with non-alcoholic fatty liver disease, or now as we know it as MESOL-D. So having known this, it is then expected that if we delete CCAM1 from hepatocyte, do we then lead to insulin resistance and steatosis? And would this lead to hepatic fibrosis? So in our latest, we use different, throughout our studies, we use different models of mouse targeting CEACAM1 expression globally or in the liver, gain of function, loss of function. I'll share with you a few of them, the most recent ones. So most recently, we use albumin-3 to eliminate CEACAM1 from hepatocyte, and using evaluating C/I ratio, C-peptide to insulin ratio during glucose tolerance tests, as being done recently in patients. And we showed here, if you just look at the green versus the red, you see that spontaneously these mice without CEACAM1 in the liver manifest decrease in insulin clearance, followed by a decrease in insulin sensitivity index without affecting insulin secretion. They also develop spontaneously fat deposition and inflammatory infiltration. Again, these mice are fed regular diet. So as such, we showed that indeed impairment of insulin clearance caused by decrease in CEACAM1 in hepatocyte can lead to chronically elevated level of insulin, which will lead to downregulation of the insulin receptor in hepatocyte, hence insulin resistance, and also increase in the fatty acid synthase activity, because now you don't have anything to, you lose the pulsatility of insulin in the portal vein. So you increase fatty acid synthase activity. This will lead to packaging of triglycerides into VLDL particles to be redistributed to the adipocytes. And eventually the adipose tissue, despite their higher insulin sensitivity, they eventually will reach their saturation. And they will start releasing excessive fatty acid as well as adipokines that we all know together lead to systemic insulin resistance. So as such, we showed that impairment of insulin clearance does not only, as we all know it, play a compensatory to insulin resistance, but it can itself also be a causative role, play a causative role in terms of insulin resistance. The mice did not only show steatosis and inflammation, but they also showed spontaneous collagen deposition, as shown in B versus A. And of course, if you give them high-fat diet, this all will be more robust. But let's stick to regular diet for the sake of time. And so what happens if you take the global knockout that manifests the same phenotype as the liver specific knockout and bring CEACAM1 back only in the liver, and let's see if this will reverse the phenotype. So we took CEACAM1 knockout null mice that you could appreciate the spontaneous obesity, body increase in body weight, increase in fat mass, decrease in lean mass. And if you breed it with the transgenic mice that we have made to overexpress using a polyprotein A1 promoter to drive exclusive overexpression of CEACAM1 in the liver. And so as such, we rescued CEACAM1 specifically in the liver, as shown in these mice labeled with purple. As you can tell, this decreased body weight, decreased fat mass, normalized them, and normalized, restored lean mass to normal. It also restored insulin clearance, red versus gray, restored insulin level, and if you do a clamp analysis, hyperinsulinic euglycemic clamp analysis with Dr. Jason Kim at UMass, you'll see that these mice again developed a global insulin resistance in the liver, as well as decreased uptake of glucose in skeletal muscle, white adipose tissue, and brown adipose tissue. And when you rescue CEACAM1 specifically in the liver, you normalize insulin action and insulin response. You also, here is the null mice, spontaneously again at 8 months developed microsteatosis as well as macrosteatosis and infiltration, inflammatory infiltration. And when you rescue CEACAM1 in the liver, you decrease NAS score, and you normalize the histology. You also normalize fibrosis. There is no fibrosis. You completely get rid of fibrosis in these mice.
Sorry, she's going, probably she didn't understand that was only 15 minutes. I think that we'll put everything on demand, but we need to go on to leave the space to the next speaker. And it's my great pleasure. It's already 20 minutes. It's my great pleasure to invite the next speaker. I know it's a tense situation, and is Professor Rotonia Carr, and is the head of gastroenterology of the University of Washington, and she will talk about the role of the lipids droplets in MASH. And thank you, Rotonia, for accepting this invitation, and here it is.
Hello, everyone. Thank you so much for the very kind invitation to be here with all of you. It's my first ADA meeting. I'm so excited. I am a hepatologist. I'm going to speak to you a bit about the liver, but not only about the liver. My talk will include some general information about the role of lipid droplets as organelles in MASH, but also I'll share with you just a snippet of what we're doing in my lab. And here are my disclosures. I'm not going to go through the progression of MASH. You all are already very familiar with this, but I have highlighted in yellow two areas. The first is around steatosis. My lab has studied steatosis squarely. We say we're steatosis, not fibrosis, not cirrhosis lab. And increasingly, we have been interested in that transition from steatosis to the earliest stage of steatohepatitis. But my lab mates and I very well understand that when we talk about MASH, we're not only focused on the liver. We know that MASH is a systemic disease. It's predominantly a cardiometabolic disease. In fact, the number one cause of morbidity and mortality is due to cardiovascular disease and diabetes, for example. So most of the studies that we do in our lab also include whole body phenotyping of mice when we're looking at mice, for example. And I hope to convince you that perhaps some of this cardiometabolic phenotype, which we have yet to really unravel, has something to do with the lipid droplet organelles themselves. Here I have a photomicrograph from an older study, over a decade old, where we learned that patients who have a combined phenotype of macro and microsteatosis tend to have worse liver outcomes. On the bottom part of this figure is a table. And if you look at category 4, the highest fibrosis stage of cirrhosis, hopefully you can see from the back that there is a larger percentage of these patients who have a microsteatotic phenotype. And we have been interested in this question for a long time. Does lipid droplet size matter? And if so, why? And so what I'll talk to you near the end of my 15 minutes is a little bit about how we're hoping to draw a connection between phenotype of lipid droplets and cardiometabolic phenotypes. But first, I'm going to start with structure. I like to get back to the basics, especially when I'm talking to an audience who may not think about the liver all of the time. Just as a brief reminder, the liver has a very unique circulation. Two blood flows connect into what is the hepatic sinusoid. And this sinusoid is a specialized endothelium. It's fenestrated, so substances can go in and out of hepatocytes at will. In between the sinusoid and the hepatocyte is this virtual space. There's hardly anything in it except for some unactivated stellate cells. And this is important when we're thinking about the role of lipid droplets in MASH. I'll take you through the life cycle of lipid droplets to start. LDs start as an outbudding of the outer membrane of the endoplasmic reticulum, and through a series of maturation steps, mature into what we call a mature lipid droplet. And in hepatocytes, that lipid droplet is a core of neutral lipids, predominantly triglycerides and cholesterol esters, and it's surrounded by a thin phospholipid monolayer, and it also has, this monolayer has multiple enzymes and proteins involved in lipid homeostasis, and we and others have also shown these proteins are involved in glucose homeostasis. In red is circled the perilipin family of proteins. These are the predominant protein classes of lipid droplet proteins. And in hepatocytes, perilipin 2 or plin2 is the predominant perilipin protein, and our lab really focuses on the biology of this protein. Now, once a lipid droplet has formed, it can undergo several fates. It can continue to store long-term. It can be repackaged and secreted as VLDL particles. It can undergo degradation through either lipolysis or autophagy. But what I want you to focus on is what's happening at the bottom of this graphic. Here you're seeing a compressive effect of the lipid droplet. So imagine a hepatocyte that has a very large lipid droplet. That hepatocyte is now pushing directly on the sinusoid and causes a local ischemia, a bit of hypoxia. And it also prevents circulating immune cells from traveling safely through the sinusoid. Those immune cells will become activated, incite their inflammatory profile, then ignite the hepatic stellate cells, who then undergo a process of activation where the collagen fibrils will end up getting deposited alongside these hepatocytes. And so if you were to look at ultrastructure, here you would see at the bottom two figures a normal fenestrated sinusoid, but just to the your left, I believe, of that, or your right of that, you will see that the sinusoid changes to almost like a regular endothelium, where you lose the fenestrae and basement membrane is deposited. And this has the impact of causing higher portal pressures. Now, we may not see this clinically at the onset because patients need to have a lot of this happen before we can measure clinical portal hypertension. But please do remember that when you're looking at a steatotic biopsy on standard H&E, that this is all happening. And so I really don't like the term benign steatosis or simple steatosis because there's a lot of biology that's underlying those lipid droplet accumulation. And if you were to look on even higher power, you can see that there's deposition of collagen fibrils that happens in between the hepatocytes. It's so-called chicken wire fibrosis. And we don't have clinical tools to measure this in any given patient, but we should be very worried when any patient develops steatosis because this is likely happening. So that's the structural role of lipid droplets in MASH. Now, what about the genetic and physiologic roles? So investigators in my team some years ago looked at GWAS studies in MASLD, as did a number of groups, and we came up with 77 loci that are implicated in MASLD or MASH pathogenesis. And what's important here to know is that most of the genetic risk comes from genes involved in lipid droplet proteins or lipid metabolism. And highlighted in yellow, PNPLA3, TM6SF2, these are two of perhaps the most validated genes in MASLD pathogenesis. These genes were then used as part of a polygenic risk score by these authors and published just a year ago, where they established that we actually have at least two classes of patients who have MASLD, those who will go on to develop liver-specific complications and those who have predominantly cardiometabolic phenotypes. And we all have known this is the case. Some people, why don't they get progressive liver disease, while others are really having a lot of other sequelae. In the red are those who are in the so-called cardiometabolic cluster, and in blue are those who are in the liver-specific cluster. And what's important to note here is that they all do get chronic liver disease of some kind, but those who are in the liver-specific cluster tend not to get as much cardiovascular disease or diabetes. And when these authors delved a little bit further and looked at the genes that are specific to these different phenotypes, they found that those who are in the liver-specific cluster tend to have derangements in genes involved in lipid metabolism, and those who are involved, and those patients who are in the cardiometabolic cluster tend to have derangements in genes involved in lipid transport or inflammation or immune dysregulation. Now, when my lab sees these data, we think about the lipid droplets and the sizes, because we know that classically, when you have derangements in lipid metabolism, you're going to have a macrosteatotic phenotype. And classically, when you have more of an immune profile, again, that 2010 paper, you're going to have more of a microsteatotic phenotype. And so we are wondering now, is there a way for us to tease this apart? Because the liver disease progression, because of the structural changes I told you about LDs, makes sense. But what doesn't make as much sense is how do you get from MASLD and steatosis to some of the cardiometabolic consequences, and does lipid droplet size matter? So what I shared with you before is that PLIN2 is really a very important protein. We established and others have established long ago that this protein is required for macrosteatosis development. Here I'm showing you an alcohol diet, but this has also been shown in high-fat dietary feeding as well. PLIN2's main role is to really protect that core of neutral lipids at all costs by inhibiting fatty acid oxidation, lipolysis, lipophagy. And we have done a lot of work on the regulation of this protein, which I will not be sharing with you today, but I'm happy to answer any questions that they're also regulated by, this protein is also regulated by ceramides. Now, fortunately, we are able to tackle this question of do lipid droplet size matter and what consequences do they portend? Because a human PLIN2 polymorphism was reported not that long ago, SIR251 Pro, which is normally a very conserved area of the gene across multiple organisms. We were able to overexpress this polymorphism in mice who are in a whole body null background. Through AAV8 overexpression, we overexpressed the polymorphism or wild type and then fed these mice a high-fat, high-fructose, high-cholesterol diet over 12 weeks and asked a number of physiologic questions, liver-related questions. And alongside that, we were able to look at 3 large population databases of patients who have this polymorphism and ask a number of phenotypic questions. What we found was that this polymorphism leads to lower amounts of liver triglyceride, and that is because this polymorphism prevents large droplet formation. These mice, in this case, can get steatosis, but the droplets are really small. But the serum levels of triglyceride are normal, and the ALT is about the same. We were also interested in this trend, at least in the chow-fed state, of a higher trend toward a higher level of VLDL secretion, although that was not borne out statistically or in the FFC condition. But otherwise, if you looked at these mice, they look about the same. They're eating the same, they're weighing the same, their liver weight was about the same. But they do have a relative lipodystrophy with a lower amount of adipose tissue in the polymorphism mice. What I don't have opportunity to go in here, we also saw that these mice have very high fatty acid oxidation and energy expenditure. When we looked at RNA-seq analysis to understand what was different about these mice, we were really excited to see that our hypothesis may make sense. These mice have an upregulation of inflammatory pathway genes and, no surprise, a downregulation of fatty acid metabolism genes as compared with the wild-type condition. But in humans, this polymorphism is rare, and we also see that they have lower amount of liver fat on MRI, lower amount of visceral adiposity, but higher level of liver iron, which as a hepatologist leads me to think that their livers are inflamed. And when we look at the human phenotypes, we are seeing that these polymorphisms are indeed associated with cardiometabolic comorbidities like diabetes, cardiomyopathy, and perhaps even heart block. Now, going back to that Nature paper with the different phenotypes, we were interested to see that they looked at multiple metabolites, and sphingolipids was one of the classes that they looked at, which again is one of the areas of my lab's research. And they found that these differences in metabolites impact lipid processing, and metabolism, and even an inflammatory profile of these patients. So when we looked at our own mice and compared our data to theirs, we were excited to see that we found some of the same things. Here I'm showing you ceramide levels, and what we know about ceramides is that the ratio matters. Very long-chain ceramides are thought to be so-called protective, and long chains are more deleterious. And that ratio, the long-chain to very long-chain ceramide, has already been established to predict both cardiometabolic and even diabetes or pre-diabetes risk, which I'll show you in the next slide. And here I'm showing arrows showing that our mice have lower levels of very very long chain ceramides as compared with long chain ceramides. And here's just a study showing that this ratio matters in your world as well, in pre-diabetes in this study. My last slide that I will share with you, and 15 minutes that I have, is that this cardiometabolic phenotype we suspect may also have something to do with the VLDL particles, and not necessarily the rate of secretion, but how large they are and what they are carrying. So here in humans, in those data sets, we were also able to ascertain that those who carry the polymorphism have larger VLDL particles, and importantly, they're carrying cholesterol esters and cholesterol. And in the cardiology literature, I'm now getting a little more up to date on that, we understand that perhaps it is this VLDL cholesterol containing particles that account for as much as half of the cardiovascular risk, maybe more than LDL as we once thought. So this is very interesting to us. We have ongoing studies in our mice predominantly to understand this cardiovascular profile. So in summary, MASH has at least two distinct phenotypes, liver and cardiometabolic. Lipid droplets play an important structural role, phenotypic role, and genetic role. PLIN2 is required for normal LD homeostasis. Too much PLIN2 is pathologic, but so too is aberrant PLIN2. So strategies that just knock out your ability to store lipids is probably not strategies that I would ever support. Microsteatosis we think is important, not just by correlating disease risk, but maybe even involved in the pathogenesis. And unraveling this biology can help us gain insight into new therapeutic options for this condition. Thank you.
Very much. We decided to, I think, we can have the question for this speaker now, and instead of waiting. And so if there are any questions, in the meantime, I have a question for you. I don't know if can you hear me? Yes. I don't know if you have seen the paper by the Professor Mingrone, because she has found that the circulating PLIN2 correlates with the amount of PLIN2 in stellate cells and is also associated with fibrosis. So I was wondering, since you found this association with ceramides, if this could be, mediated by ceramides. What do you think?
Of course we think that. Ceramides are really a very provocative lipid species because they can signal, right? And they impair insulin signaling, for example. They promote apoptosis, and cardiology even have a role in promoting atherogenesis, promoting apoptosis of the cardiomyocytes. So we are very interested in the role of ceramides per se, and we have landed on one particular ceramide enzyme that we think is critical, and we are happy that other groups have come up with the same one, that's ceramide synthase 6, that produces long-chain ceramides. And so we have a number of studies in our lab really trying to understand this biology better. So yeah, I do think there's something there. And we don't study hepatic stellate cells because we study steatosis, not fibrosis, not cirrhosis. But I've always been interested in the lipid droplets of stellate cells, because once they become activated, they lose their lipid droplets. And so I'm happy to see that there are others who are working on that piece.
Please.
Hello, I'm Petra Hirsova from Mayo Clinic. Excellent presentation, very insightful. I want to ask about the macro versus microsteatosis and during disease progression. So if we see more microsteatosis in later stages, do you speculate it is because during progression the macro changes to micro, or you think maybe patients that start with micro are more likely to develop fibrosis?
That's a really great question. We don't know the answer to that, but I would say the preponderance of sort of clinical epidemiologic studies, we rarely see only microsteatosis. We usually will see macrosteatosis at early stage disease and or a mixed phenotype. My suspicion is that, at least for patients who don't have aberrant polymorphisms and are able to develop large lipid droplets, that those lipid droplets do change over time. And what drives those changes, we don't know, the initial inciting event, but they move from a ability to safely store neutral lipids to an aberrant phenotype, where now we have bioactive toxic lipids circulating in hepatocytes, causing inflammation, and upregulating these really kind of oxidation pathways. So I think that is what is happening, and then that is what drives fibrosis.
Thank you.
Thank you so much. Yes, we can go to the next speaker is Professor Yangur Rui from Professor of Molecular Integrative Physiology, University of Michigan. He will talk about liver cell heterogeneity, plasticity, and dynamics in MASH progression.
To open it?
Maybe with the
Maybe too
mouse.
fast?
Yes. No,
Oh.
I think here, start.
Oh, okay.
Yeah, it's a
You're
quite
smart.
complicated.
Thank you.
You're welcome.
Thank you. Thank Yangban and the AD organizer to provide me this opportunity to share some of our data, recent data. And Dr. Naja and Dr. Khan give a beautiful talk about the metabolic pathways in the liver disease and the lipid metabolism, lipid droplets. I'm just wondering what else left over for me to talk about it. So now, today I'm going to use one of the genetic models to illustrate that and hepatocytes not the same. Besides the zone 1, zone 2, zone 3, we are known textbook knowledge, and also it's extremely heterogeneous populations, and not only regards of metabolic pathways, sometimes it's plastic, even goes beyond the functional heterogeneity, goes to change the cell fate, like the cholangioscytes. So now I'm going to use one of the simple genetic model that's a TREF2 genes. And TREF2 genes is adapter molecules well known in the inflammatory pathways and forms a complex connects to a receptor to the downstream NF-kappa B pathway. Now this one is less known, less well known. It's called a non-canonical NF-kappa B pathway initiated by the kinase called NIK, NF-inducing kinase, which binds phosphorylated IKK-IVA, which phosphorylated this component P100, causes cleavage forms a mature transcription factor, goes to nucleus and initiates gene expression, therefore leads to the immune response. So this pathway, this kind is a key regulator. Normally expression level is very low, and why is low? Because it's constantly degraded from this complex. This is the E3 ligase, and recruited to the NIK kinase through this TREF2, TREF3 complex. In response to the cytokines, other stress stimuli, and then you disrupt this complex, NIK is accumulated and activates this pathway. So this is a cartoon of the TREF2, and then I have NIK usually binds to this is the protein-protein interaction domain. So why we're interested in these molecules, and just look at the NASH models. We use different NASH diet. The common finding is that the TREF2 level is higher after induction with NASH diet, and now increase. Well, first, I think maybe have their pathological contributions, and why not we just knock out to prevent the increase? What happens to the liver? So we generate liver-specific knockout. Use the classical albumin-3 plus the NIK TREF2 flux. Indeed, and TREF2 is knockout because TREF2 is an active regulator. You knock out this one, you activate this one. We use the assay to measure the abundance of the P52 is highly activated as expected. But NIK also functions as the MAP3K, activates MAP3 kinase. You can see MAP kinase will also activate if you remove the repressor. So what happens to the liver? And we premise in a short term, 4 weeks high-fat diet, and got a quite significant liver injury marker is LT. That's a marker, plasma LT, liver injury marker. And when you look at the liver histology, fatty liver, there's not too much change. But you do see increase in the cell death based on tunnel assay. You do see the increased inflammation, and another remarkable thing is that the increased liver fibrosis. Now, Western blot and show more quantitatively that this is the FSMA, that's a marker for the hepatic stellate cells activation is highly increased if you delete TREV2 activates NIC in the hepatocytes only. And this cell death molecule is highly elevated. So that's a genetic model, and now we also use the adult onset deletion because we fed mice, use the TREV2 increase, and then we ask the question after increase, we delete them, use the CRE-dependent deletion. What happens to the mice to make the mice better or worse? And it makes the mice worse, even though the fat content is similar and then got increased oxidative stress and increased cell death, increased liver fibrosis by multiple assays. And Western blot confirms the liver injury, cell death molecules increased by the deletion of the TREV2, and this fibrosis marker increased in the deletion mice. So this is kind of liver injury fibrosis. It seems to be the last common feature for multiple liver disease, in addition to or beyond to the MASH, like drug-induced liver injury and alcohol-related liver disease, and similarly, and TREV2 seems to be involved in all of those pathology. Now here we use a CCL4, mimic drug-induced liver injury. If you knock out TREV2 in hepatocytes, and the fibrosis is markedly enhanced in the mutant mice. Now we do the opposite way, and if we overexpress TREV2, can we have the protective effect? The answer is yes. The mice liver is much more protective against CCL4-induced liver fibrosis. Now, that's a drug-induced, and now we goes to the MASH model that use diet-induced MASH, overexpressed TREV2. We don't see too much, just like knockout in the lipid droplets, lipid content, but we do see markedly improved in the liver fibrosis. So now, let's go, we knock out in the hepatocytes, and then we ask that every hepatocyte behaves similarly or different, seems to be different, based on the phenotyping. And now we do the single sequence analysis to analyze different cell populations. And two populations stands out quite obviously. One is the liver inflammation, macrophage highly increased in the cell number. Another population is the cholangiocytes, is increased by several fold based on the single cell sequence analysis. Then we confirm that by immunostaining. In the knockout mice, use the cholangiocyte specific marker CK19, and as you can see in this, for 4 weeks half a diet, and you see massive increase in the bioduct epithelial cells, cholangiocytes. And we use additional marker SOX9, which always also marks the bioduct epithelial cells is highly increased compared with the control. And there's no gender difference. Males and females similarly have this response. So now this newly generated cholangiocytes is largely enriched in the zone 2, and we still see basic zone 3. That's a zone 3 marker, and this is zone 1, and then increased cholangiocytes. That's a green. That's a cholangiocyte marker is largely in close to zone 2, zone 1, but not to the zone 3. But the liver still maintains the basic zone structures. And this is another model that is a genetic embryonic onset, and here we just like to delete the TREV2 in adult mice to see we can recapitulate the phenotyping. Indeed, you put mice on a high-fat diet, cause metabolic stress, and you inject it with TBG CRE AV virus to acutely to delete the TREV2, the TREV2 flux mice. And you're only waiting for about 4 weeks, and you look at a massive increase in the cholangiocytes mass. in the liver field is called a ductular reaction, and another marker shows similar things. Now, considering this one and a NASH diet have increase of the TREV2, we think the increased TREV2 may serve as a physiological protective mechanism to maintain the hepatocyte's identity function. If you do not have this one like a knockout, the hepatocytes goes crazily and de-differentiation and transdifferentiation. And this kind of the time dependent as expected, and using knockout mice and use the TBG create to acutely to delete TREV2 in the liver, in the hepatocytes, and now 2 weeks you have modest increase in the cholangioscytes, and by 4 weeks you have obviously massive increase in the ductular reaction. And now we stain the liver sections also with the hepatocytes marker, and we find a population of the cells. This population increase in the time dependently is double positive, express both cholangiocytes marker as well as hepatocytes marker. We think that this population represents the transition from the mature hepatocytes to the cholangiocytes, and because of you lost the gatekeeper TREV2 in these hepatocytes. So now provide additional evidence that it's really the transdifferentiation instead of the proliferation of the existing cholangiocytes in the knockout mice. So we use this AAV lineage tracer tools, and we injected 2 viruses simultaneously into the mice. One is TBG to create to mark the hepatocytes. We also inject the reporter M-cherry. Expression of M-cherry depends on the create, means mark the hepatocytes with M-cherry red. Then you delete TREV2, you inject this virus into the TREV2 flox mice. Therefore, this create delete TREV2 at the same time. And you can use the wild type as a control, right? So now you can see that you can see quite a significant increase in the yellow color. That means that this cell from the hepatocytes, because they are red, then they express cholangiocyte marker. That's the CK19 green. So this is a typical lineage tracing approach to show that this newly generated cholangiocytes indeed is born from the mature hepatocytes. Now, this is another model, just written a long time. You got a massive cholangiocytes proliferation generation. You see more. So put them together like this way that the TREV2 normally keeps the hepatocytes identity function. If you don't have them, hepatocytes and have less ability to keep their identity and tends to de-differentiation and transdifferentiation to the other cell type, particularly the cholangiocytes. Now, what is the long-term consequence if you mess up that mechanism like TREV2 to maintain the hepatocytes identity? You can imagine that, well, goes to the metastasis, the tumor genesis, indeed, and we use chemical-induced liver cancer model, the wild type. We give it a short time. Usually, this model take about a long time, over 10 months or a year. You have liver tumor. Now, 18 weeks, we see massive liver tumors in the knockout mice, and you barely see anything in the wild type controls. Now, indeed, this tumor, you do histology, is kind of the mixed tumor. Some of them hepatocyte marker, and many of them express the cholangiocyte marker. That represents the transdifferentiation and gains some additional mutation and becomes the liver cancer cells. So this is a mixed cancer, liver cancer. That also happens in the humans. So put everything together, and this is the working hypothesis. We still have a lot more work to do. We think that the TREV2 is kind of serves as a gatekeepers to maintain the hepatocyte's identity to prevent the de-differentiation and transdifferentiation. What is the downstream pathway after TREV2? NIC certainly is one of them, but it's not the only one. We have a couple of additional candidates, which is and keeps the hepatocytes unstable tends to differentiate it. We have to have a blocker to block them. Otherwise, they go crazily and it goes to the cholangiocytes. But if there are some pathological factors that weaken this weak gatekeeper's function, now goes this pathway and dedifferentiation, transdifferentiation, and ductular reaction, and which this is not newly generated hepatocytes or cholangiocytes or these double positive cells. We collect the cognition medium from them. They contains a lot of the cytokines that promote liver inflammation. And they also promote the stellate cells activation and contributes to the liver fibrosis. So those, even though initially the population is small, they grow and they produce like endocrine cells or paracrine cells. The secret mediators affect the microenvironment. So over the long time, which is contributes to liver cancer. So finally, I would like to. I have a lot of people to thank for besides the NIH and the ADA to support my career, and lab people, like current and former lab members, did a tremendous and beautiful work for moving science forward. And today I mainly do postdoc and Qing and Ziguo. Did most of the work, and wonderful collaborators, particularly engineering, my colleague in the UFM. We just collaborate with many things in the liver. Thank you so much. Welcome to any questions.
While we're waiting, I wanted to ask you, do you know any drug that is affecting this molecule, this gene TRF2?
TRF2 is a E3 ligase itself. It's a dapler proteins, initiated protein complex. Now, in the NASH model, we see increase in the expression, and some inflammatory cytokines can cause the transcriptional activation of these genes. And these genes also can be mobilized to different, like to the ER or to the other different part of the plasma membrane. They call it sequester the proteins and then relieve the downstream molecule like need to be activated. And regulation of this TRF2 is a kind of multiple way, like other.
Yeah, because you showed that the ablation of the decrease in function is associated with liver.
Right.
Yes. So,
Yeah,
do you think it's initially a protection function?
Yeah, that's
going
exactly
up?
as you said. Initially, liver trying to keep the cell, trying to regulate the TRF2, but have the limits.
Yes.
Just like insulin resistance, we pump more insulin, but we have limits in the beta cells.
Of course.
Yeah, I guess the TRF2 breaker have the limits.
So, and do you know if any of the drugs that are now currently on investigation for the treatment of MASH will have some effect? Yeah.
We're trying to in the process to get a patent for a compound. But hopefully.
Okay, of course, everything is next time. Okay, thank you. Question
Great.
there.
So I wanted, maybe I could pose it to the two speakers, both?
Yes,
perhaps?
of course.
Yeah.
Yes.
So my name is Sunil Koliwat. I'm from UC San Francisco. And great talks. I'm wondering whether from the standpoint of PLIN2 and the mediation of lipid spillover and inflammatory activation, or from the level of TRF2 upregulation, whether you've seen a specific zonal pattern of distribution with nascent steatosis, that the protective effects, if you will, of increasing neutral lipid storage or increasing TRF2 to maintain identity is specifically manifest in particular regions of the lobule, and whether that matters. I'm just thinking that these inflammatory mediators probably are released in certain areas and diffuse from there, and you see inflammatory activation is not evenly spread equally across the lobule, but begins in certain areas. And I'm just wondering whether TRF2 or PLIN2 reveal something about where the signal emanates from.
I can answer that. Not for PLIN2 because it's a structural lipid droplet protein. it itself isn't going to really have a signaling opportunity. But we have noticed with ceramide enzyme, ceramide synthase 6, that the first place that it's expressed is in zone 3 hepatocytes when there's very little lipid droplet accumulation. That was in an alcohol model. So I do think that there's something there.
And what about the localization of PLIN2 in terms of its localization with respect to the ER or mitochondria within the cell? Is that zonally different?
No, PLIN2 is always adherent to the lipid droplet organelle.
Right, but the droplets themselves are localized, closer to or in apposition to other organelles.
Yeah, they're
depending on the status of the cell, right?
Yeah, but if you were to look at ultrastructure, they're all together.
I see.
The ER is adherent to the LD, is adherent to the lysosome, and so it is really challenging to tease that piece apart. Yeah.
And I guess for TRAV2?
I can address the TRAV2 related zonation things. And based on the histology, I showed briefly that I think the TRAV2 function to keep the cell identity, not can keep every cell's identity, only subpopulation. If you delete the TRAV2 and those populations are affected more dramatically, those populations close to zone 1 and zone 2, but not is far away from zone 3. And we also did a sequence analysis and we found another population which tends to call the disease-associated hepatocytes. They not only have the feature of a couple of the gene expression pattern, those also enriched in the zone 1, zone 2, but in the MASH models. And that's just the initial hypothesis. We call the terms the disease-associated hepatocytes because that's unique. And we search the single sequence, cell sequence analysis that's also observed in other NASH model or MASH model, even in the human MASH model. So do have some kind of the feature about separation spatially.
Great, thank you.
Hi, Petra Hirsova from Mayo Clinic. I have a question about TRAV2 this time. I'm curious, so it's adapter protein that's recruited to TNF receptor superfamily and maybe some other receptors. Can you speculate in this case, what is the upstream receptor or what's the signals that are maintaining the differentiation or survival of these hepatocytes?
And I speak, given that TRAV2 is very documented involved in the inflammation response, I think upstream most likely a family of cytokines or multiple family of cytokines act on the upstream. It's not like at the initial phase. It's only after steatosis, during the progression, liver have kind of inflammatory environment. Those kind of cytokines or inflammatory mediators somehow engage the TRAV2 and initiate those downstream pathways. So, need additional work. Thank you so much for your question.
Well, if there are no further questions, I will close this wonderful panel discussion, and I'd like to thank the speaker for their talks and for the lively discussion, and thank you very much also Professor Yang Bum for the organization of this symposium. And with this, we close and we wish you a wonderful rest of the meeting. Thank you very much.